And you can start. Yeah.
Yeah, so welcome to this session. We've got 3 exciting high school teams coming up, and East Coast Bio Crew is going to be our first one. So please kick it off with your 5-minute update.
Should I unmute?
And yeah, please present slides in the Zoom and speak into the microphone so that our remote judges can see as well.
Who's presenting the slides though?
Tiny bit of debugging, which is fine.
Do I have to log into Canada? Oh.
You will use this microphone, so you don't need to unmute your laptop. Okay.
Is it this one?
No, I need the slides.
Yeah, these are the slides.
Oh, gotcha.
See, those are right.
Ready? Hello, judges, and welcome to our 5-minute update. We're so grateful to be here, and we're going to start with an update of our current work.
As a recap, we want to tackle phosphate pollution by introducing 2 pathways in a soil bacteria known for its root nodulation. Over the summer, we were able to successfully introduce genes into Rhizobium. Specifically, we inserted a pstSCAB operon that would code for a high-affinity phosphate transporter. More notably, we introduced a PQQ GDH pathway that would code for an enzyme and cofactor that could solubilize mineral phosphate into a usable form. We were able to confirm these genes were inserted in Rhizobium by performing PCR and running a gel.
In addition, we have some evidence that at least one protein might be expressed. This would be the GCD gene that codes the enzyme glucose dehydrogenase. You can see the band in the image. You have to what? Oh shit.
Hey guys, we don't see any screens or yeah, okay, there we go.
Oh yeah. For our phosphate uptake assays with agar plates, we saw promising results after we changed our media we used for testing. In the image, you can see halo-like clearings that encapsulate bacterial spots indicating phosphate uptake. For our solubilization assays, we saw promising results as our engineered bacteria had markedly lower concentration of phosphate in the solution. Not only that, but we engaged with a diverse set of stakeholders and used their feedback to optimize our project. Lastly, we created educational materials focused on diversity and science that are ready for classroom use and teacher approved.
Despite our results, we plan on continuing both the uptake and solubility assays before we have conclusive results. As of now, we plan on continuing the phosphate plate assays. Since this procedure lets us see our results, we plan on continuing it until we can hone in on the details. Mainly, we want to control the bacterial solution dilutions so that we can eventually compare the halos between sample sizes. We also want to ensure that our tests with P fluorescence are not compromised by its different living preferences. We also want to test for gene expression in E. coli to see if potential issues are species specific, since we have never worked with Rhizobium before. We also want to test for different promoters, since the one we chose is not known to work well with Rhizobium. This is because we selected a vector with a lac operon and therefore the lac promoter.
Like our phosphate uptake assays, we plan to continue with our colorimetric solubilization assays. During our initial test, we used a sample volume of 500 microliters, which couldn't be measured by the machine as it had too many cells. During our second phosphate assay, we lowered the number of cells and did receive a conclusive result. This makes sense as higher volumes with lower concentrations could lead to the solute settling, and that would incorrectly influence our result. However, we did receive a positive result during our second phosphate assay, which is really important, and you can see that our genes do influence the solubilization of phosphate. This means that our project could work. As you can see in the leftmost image, the absorbance rates for the pstSCAB pathway and the PQQ GDH pathway are significantly lowered when compared to the wild tropici pathway, the unmodified Rhizobium. We did use P fluorescence as our positive control, and this makes sense as the absorbance for our positive control is much lower when compared to our pstSCAB and PQQ GDH pathways, and this still means our result is valid because These pathways are both lower than the wild type rhizobium. The last thing that we want to do is to find a way to isolate our solubilized phosphate from the solution containing the bacteria. This is because we've received similar low absorbances for both of the pathways. We expected the pstSCAB pathway to have an overall low absorbance rate because the solubilized phosphate would be taken into the cell. However, we expected the PQQ GDH pathway to have a higher absorbance when compared to the pstSCAB pathway because the mineral phosphate would be in a form that is free to interact with the phosphate reagent provided by the kit. We theorized that we received a lower absorbance because the phosphate was in its form that was ideal for the rhizobium to use for growth. The next big thing that we want to do is to repeat our colorimetric assay with all of these things in consideration that would help us have a more definitive conclusion.
We also wanted to verify.
Nearly out of time, so just.
Oh.
Yeah, I think jump through this slide quickly, just giving you a warning that we're cutting into question time now.
Oh, we also wanted to verify the expression of our genes with quantitative PCR, also known as real-time PCR. We have already extracted RNA from our engineered bacteria, and we plan to run the qPCR in the near future. If the test shows insufficient gene expression, we would try a few things. We could try a different promoter, which would coincide with modifications for the before mentioned tests, or we could try a stronger constitutive promoter to maximize gene expression. In addition, we can try adding more of the, is that it?
We will explain more during the Q&A, if that's okay.
Yeah, sorry to cut you guys off. But we're pretty much at 6 minutes now, and we want to make sure we have time to ask you questions. So anyone who would like to answer questions, please come up towards the mic, and we'll give our first chance to answer questions to our remote judges. The way we're going to do question asking in this session is we'll try to write our questions in the Zoom as well, just because that's more accessible for across languages. And then we'll unmute and ask our questions verbally as well. So remote judges, do you want to jump in with questions first? Yeah, could we check the allow participants to unmute? They can see the slides now. That was from earlier.
Yeah. Can you hear me?
Okay, there we go. We can unmute. Yeah. Oh,
Great.
Rocky, do you want to go first? You can go
Oh,
first.
you can go first, Dennis, as well. It's a sec. It's fine.
Okay. Okay, great. So I just typed in sort of the crux of my question is, so on your wiki, guys, you had this gel with your protein, looking for expression. And I think you mentioned 87 and 95 kilodalton band. And I'm wondering if you could talk a little bit more about what you think that band actually is and whether or not you think you saw it in one of the earlier lanes.
Hi. So I think I know what you're talking about. In terms of the 87 kilodalton band, that was supposed to be coding for our glucose dehydrogenase enzyme. And what we did is we had the first column be a control. So we compared it against the control to see if we could see any new bands. So around the 87, or it wasn't indicated on our actual ladder, but between 75 kilodaltons and 100 kilodaltons, there appeared to be a new band on the last column, which would have had the PQQ GCD genes inside. And then referring to the 95 kilodalton, because one of our genes that we inserted was pstSCAB, which contains the pstABC transporter, those proteins that are produced have a high association, because they're making the transporter. So we thought it was very possible that these proteins could be associating with each other in the protein gel rather than being fully denatured and not associated at all. And so we were aware there was a possibility that we would see larger proteins than each individual.
Did you guys consider how you might follow up to confirm the identity of the protein or proteins in that band?
So I think we probably would just want to run another SDS page because we that was close to the end of time. Our first protein gel, we couldn't really conclude anything because the gel broke. So it was very hard to see what was going on. And so we ran it again. And I think just overall okay. Long story short, lots of our experiments, it was very difficult to do, not only because of how often our meetings were, but just a lot of us on this team are very new, and so things happen. Human error is something that we are trying to learn from. We're trying to solidify these protocols, and so I think if anything, the best way to verify this is just to try it again and ensure we know what we're doing.
Okay,
But
great.
yeah,
Yeah, no worries. Thank you so much.
Shall we jump to Rohini's question? And thanks, Dennis. Rohini, do you want to unmute and ask a question?
Okay, many thanks, team, for your presentation. I just wanted to know your take-home messages from your iGEM journey or your favorite experiment in your whole journey for the iGEM project.
Hi, yeah, I would say that's actually the nodulation of our showy tick trefoil. We conducted a series of plant experiments in order to determine what the native plants that actually work with our rhizobium is, and we found nodules on our showy tick trefoil. So that was a big highlight.
Another big highlight from the Genspace group was our transformation of the rhizobium. A lot of us got to use electroporation for the first time, because normally we've just done a regular Golden Gate assembly, but for the first time we got to use the electroporator, which was super duper fun. And in general, I think that our takeaways are that this is a really big learning experience for all of us. Most of us are really new to SynBio and the whole concept and the whole world of SynBio. So the fact that we're actually able to get our hands dirty is a really important thing for all of us. And I think we're super duper grateful for having that opportunity. With that being said, I think it's also important to note that science does take time and not to be discouraged by any bad results as kind of feedback for ourselves and for our future teams.
I have a follow-up question here. So Genspace, could you explain to me what Genspace is and how it's related to your work?
So actually our team was made up of two different community labs. So our first community lab was Bugs, which is the Baltimore Underground Science Base, which is in Baltimore, Maryland. And our second community lab is Genspace, which is in Brooklyn, New York. So we had two different groups and two different groups of high schoolers meeting in two different areas on the East Coast, which is actually where our name comes from, the East Coast Bio Crew. So we did run similar experiments at both of the locations, but they did very little. Like the liquid assay was done at Genspace and the plate assay was done at Bugs. But both of the groups actually did end up transforming the rhizobium and growing the rhizobium, making the media, and all the things that went into getting to the phosphate assay.
Shall we jump to Frauke's question?
Okay, thank you. I was wondering, when you did your plant experiments, you were actually seeing only nodules in your trefoil, but not in the legumes, although the rhizobium should be or is suspected, or that's what you were stating, is specific for legumes. How do you explain that?
Yeah, so actually, classically with these kinds of experiments, they're done in a much larger area. And so the soybeans and the common beans, for example, which are the ones that are actually known to work with our rhizobium, they are typically grown in a larger area and they have very controlled environments. The trefoils are much smaller, and therefore they actually fit in the greenhouse that we had. And so they had the best chance to actually make it through.
I'd like to follow up on that question or that answer and say that when we observed those results and we didn't see nodulation in the legumes, we did know that that was very unexpected. So we contacted Dr. Sharon Long, who's done a lot of characterization with rhizobium nodulation. And she told us a lot about monitoring pHs and nutrient intake in plants that we hadn't been doing before. And so we changed some of our experimental design in response to her input.
Okay, thank you.
Thanks so much. I want to make sure the remote judges have more time to ask questions if you want to jump in. Rohini, do you want to ask your next question?
Sure. How will you make sure that the plasmid that you have introduced in rhizobium will not be lost given the selection pressure is taken away, the antibiotic selection pressure is taken away when you want to go with this rhizobium in the soil or a field setup?
So to answer this question, for us, we just decided we'd start with plasmid expression because it's a simpler, but it's also, we kind of wanted to show a proof of concept first that, introducing these genes would be able to uptake more phosphate and solubilize more mineral phosphate. In terms of like implementation, if we were to actually put this as a biofertilizer, we would look more into like genomic expression, so we wouldn't have to worry about our plasmids being lost. So yeah.
Okay, thank you.
I guess that's, I'll maybe ask a follow-up question that I'd put in the chat earlier, which is, if you think about that real world application I saw in your human practices, you did talk to some of the companies that are making bacterial fertilizers. Do you have a sense why the phosphate bacterial fertilizer is such a gap right now, and most of the companies are focused on nitrogen?
So one of the big things is that, nitrogen and phosphorus, both of them are, I would say, roughly equally important in terms of importance and kind of how they affect the world. But for nitrogen, I would say that nitrogen has a higher effect on crop output, but phosphate has a much higher impact on our environment. And so by tackling phosphate, there's not exactly a sort of a field of entrepreneurship that we can pursue as well there. And so we really want to focus on sustainability. So that's why we have chosen phosphate, and we think that'll do it better. Nitrogen is very important, but we think that it is more commercial.
Just to follow up again, did you get to talk to farmers or people that work with?
Yeah, we actually talked to a few different groups of people. So we talked to farmers, and we got to talk to 2 farmers. We talked to Claggett Farms, which is connected to the Chesapeake Bay Foundation, and that was really good for us because they know a ton about sustainable agricultural practices and crop rotation and things of that nature. We also got to talk to Beth Kasuke from Hog Run Farms, and both of these farmers kind of expressed the concern that things need to be profitable to be used. So when we were talking to farmers from Claggett Farms, they were saying that farmers may be less inclined to do something if it isn't going to be profiting their numbers for them or padding their numbers. So this kind of led us to think about how we can actually not only use our project to help the environment, but to actually help crops grow and to increase crop output, which we got through the process of phosphate uptake. And then back to the other question that was asked, we did talk to Pivot Bio, which was the company that we got to talk to that we were really grateful to speak with as well. And we also learned a lot about the regulatory process from them.
And also, just one thing before any next question. When talking about the actual economic impact, the long-term impact of our project in terms of like a social, environmental, and economic sort of field is that nowadays, a lot of our fertilizers come from very unstable regions in Eastern Europe. As you know, there is conflict happening there right now, and that can cause fertilizer prices to skyrocket. And so by implementing this project, we can actually reduce that cost over time and turn fertilizer into more of a safety net than a necessity.
Thank you. Shall we jump to Dennis, your next question?
Yeah, we can. So sorry, guys. Okay, can you guys talk a little bit about how you engineered your rhizobia? Because you mentioned you transformed it, but I was trying to understand what process or processes you used for that.
Yeah, so kind of like in your question, we actually did make electrocompetent cells. So we washed them, we got rid of all of the other salts and solutes that might mess with the electroporation, because we don't want the electricity to make it's just not good if we have those extra solutes in there. So we washed them, we made electrocompetent cells, and then in terms of designing our plasmid, I'm not gonna lie, we did have some issues with that. Oh wait, is this, oh, it is not, okay, sorry. It looks like it's muted. Are you guys able to hear me?
Yeah, you're coming through this microphone.
Oh, okay, got it. Sorry about that. No, so when we were making our plasmids, we had some issues because there was a high GC content, especially when we were trying to make our glucose dehydrogenase enzyme. So instead of doing like a normal restrictive digest, we actually did PCR to clone the plasmid around the multiple cloning site, and then we assembled our genes that way. We made our electrocompetent cells, inserted our plasmids, and then we did blue-white screening with kanamycin as the antibiotic to kind of selectively choose which colonies actually contained our plasmid. So does that kind of answer your question?
Yeah, it does. Thank you.
Okay, thank you.
Thanks so much. So we have about 5 minutes left for questions. Natalia, did you want to ask one?
Yeah. Right. So you did a lot of work characterizing the best medium to grow Rhizobium, right? So exploring a little bit on that, how many repetitions you did for the medium experiments?
So the main focus of the models that we did for this project were characterizing the optimal growth rate for Rhizobium. And we did that considering different mediums and temperatures at which we can grow it. We did around 5 replicates of different media experiments. However, some of them, the data were not conclusive, or we didn't have enough of it to draw accurate conclusions. From the models where we actually had enough data that could draw conclusive evidence, I think we had about 2 replicates of conclusive trials that proved that of our original 3, yeast mannitol with low calcium was the most effective. We only had 1 trial that showed that tryptic soy broth was more effective than yeast mannitol. But if we continue in the future, we could run that again to confirm that result.
Thank you. It looks like Fokke had another question about what is your target crop?
So one of the great things about our project is that the Rhizobium is not restricted to only one species, and it actually can work with a variety of different species. So one of the reasons we have actually used like the trefoil oil for an example is because it's a plant that is native to our area. And so we hope that by using plants that are native to different areas, we can really tackle the issue on a global scale eventually.
Nice. Yeah, I saw that you managed to make the symbiotic association between the trefoil plant and the Rhizobium, yeah? And I wanted to ask you, you said you also did a solubilization assay of the Rhizobium, and this was with the Rhizobium extracted from the symbiotic relation, or the one that you already had?
So we actually used the Rhizobium that we had already transformed, so it wasn't from the symbiotic relationship.
Okay. Yeah. And how did you how was it, the assay?
Yeah, so the solubilization assay was done at Genspace, and we used a kit provided by Sigma Aldrich. Basically, how you do it is we used a spectrophotometer to emit a 650 nanometer wavelength red light, and the color produced by the bacteria would be opposite to that color. You want to add, Sophia?
So like included in the kit, there was a chromogenic complex. It's like a phosphate reagent, which would bind to free phosphates only. So the idea was that once it was binded to our free phosphate, it would kind of turn like a bluish green color, which is on the opposite end of the 650 nanometer wavelength. So when we use the spectrophotometer, we could measure the absorbance, and that absorbance would be directly proportional to the amount or concentration of phosphate in our media. And so by showing that our engineered bacteria had markedly lower absorbance and thereby less phosphate in the media, we could conclude that, for one, pstSCAB was likely uptaking more of that phosphate. And in terms of the PQQ-GDH pathway, which we weren't expecting to see lower phosphate levels, we were thinking that it's possible if our mineral phosphate was being solubilized, now we have phosphate that is free for the bacteria to use. So now instead of this phosphate binding to our chromogenic complex and turning the media green, it was being used by the bacteria to grow. And so that could explain why there were lower phosphate levels in that assay.
Just to add on to like our experimental design for the solubilization assay, everything was done in triplicates. So the averages that you saw on the screen before were the averages of the 3 triplicates to ensure more accuracy in our solubilization assay. Additionally, the Rhizobium that we used, we transformed 2 sets of Rhizobium. There's one set at Bugs and one set at Genspace that we had transformed. However, only the one at Bugs was sequenced. And for our solubilization assays, we did have the Rhizobium shipped from Baltimore to New York City. And we did that to ensure that we were comparing the plate assay and the liquid assay in a more accurate manner.
Thank you.
Thank you. All right, we might have time for one more extremely speedy question. Any remote judges want to jump in with this? Otherwise, I think Natalia is saving one up. All right.
Yeah.
Lightning round.
Right. So I saw that your assembly, it's only compatible with RFC 1000, yeah? So how do you plan to change this to make it compatible with more other type of assemblies in the future.
So when we were making the parts registry, we did see that, and some of the members who are returning iGEMers like informed us of this. And so at the time, since we had already kind of used those genes and used that construction, we didn't in short, we really didn't get the time to update it, but in the future we will try to, because I think it's mostly just like that restriction enzymes that we're trying to, so we would just try and look at the different compatibilities of restriction enzymes we could use that are RFC compatible. And so that would be how we adjust that.
Yeah, thank you. And our time is up.
Yeah, so thank you so much. Yeah, I just want to say really impressive project. Really liked how much you guys took a sort of bioprocess engineering perspective on it as well. That was just really, really fun to see in your presentation and in your wiki. So yeah, thank you so much. Great job.